Hello everyone, today we begins with the platelets. Platelets are also known as thrombocytes, where the word consists of thrombo means clot and cyte means cells. So the cell which helps in the clot formation, these cells are known as thrombocytes or platelets. Size 2 to 4 micrometer, volume 5.8 micrometer cube, shape and color, it's a colorless, spherical or oval or discoid in shape. On the Leishman staining, it looks a faint bluish cytoplasm with reddish purple granules. Nucleus is absent, so platelets are non-nucleated cells. So this is the ultrastructure of the platelet. This is the plasma membrane or the cell membrane, dense granules, closed tubular system. This is the open canaliculi. This is the granules, and this is one is the mitochondria. Now we are going to see the difference. What is the difference in the structure of the platelet than any other cells? So first of all, cell membrane. The cell membrane having the glycoproteins which prevents adherence of platelets to the normal endothelium, accelerate the adherence of platelet to the collagen and damaged endothelium in injured blood vessels. Second one is the phospholipid, contain platelet factor 3 which plays a role in the blood clotting process. Receptors for combining with specific substances like collagen and fibrinogen. Precursors of various substances like thromboxane A2. Prostaglandins, leukotrienes and platelet factor 3 and 4. Microtubules for the maintenance of a shape. Cytoplasm. Cytoplasm consists of the endoplasmic reticulum and Golgi apparatus which synthesize various enzymes and store large quantities of calcium. Second one is the mitochondria. These are capable of forming ATP and ADP. Alright. Now the third one is the contractile proteins, include actin, myosin and thrombostatin, responsible for the clot retraction. Next one is the other proteins present in the cytoplasm are fibrin stabilizing factor, platelet derived growth factor, von Willebrand factor, and granules and enzymes are also present in the cytoplasm of the platelets. So I just mentioned about the difference. What is the difference in the structure of the platelet than any other cells? Formation of the platelets. So the stages or the steps in this platelet formation. First of all, as we already know that every cell synthesis starts with the stem cell or hemocytoblast or pluripotent hematopoietic stem cell PHSC, which later it converted to the megakaryoblast. This megakaryoblast converted to the promegakaryocyte, promegakaryocyte converted to the megakaryocyte, and megakaryocyte give rise to the number of platelets. So this is the developmental pathway in the formation of the platelet. So first it begins with the stem cell or hemocytoblast, which is converted to the megakaryoblast. This megakaryoblast converted to the promegakaryocyte. This promegakaryocyte converted to the megakaryocyte, and megakaryocyte gives rise to the number of platelets. So these all are the platelets. Properties of the platelets: adhesiveness, aggregation and agglutination. So these three are the properties of the platelet. First one is the adhesiveness, second one is the aggregation, third one is the agglutination. Now the functions of the platelet, most important for practical viva as well as theory viva. First one is the role in the hemostasis, role in the clot formation, role in the clot retraction, role in a repair of injured vessels, role in defense, and transport and storage function. So these are the functions of the platelets, which is the most important. First one is the role in the hemostasis, second role in the clot formation, third role in clot retraction, Fourth, role in repair of injured vessel. Fifth, role in defense. And sixth, transport and storage function. Alright, so these all are the functions of the platelet. Normal count and variation. This is also the most important point. Normal count 1,50,000 to 4,50,000 platelets per cubic mm of the blood. Whenever some examiner asking you about the what is the normal count of either RBC, WBC or platelet, you must need to speak the whole length of this sentence. For example, if for the RBC count in male it is around 5 to 6 millions RBCs per cubic mm of the blood, like that. For the normal platelet count it is around 1,50,000 to 4,50,000 platelets per cubic mm of the blood. So you have to speak the whole length of this sentence. Alright. Physiological variations. This platelet count varies according to the age, sex, after meal, after severe exercise, at high altitude, all the things. And pathological variations. First one is the thrombocytosis. Whenever there is an increase in the thrombocyte or platelet count, the condition is known as thrombocytosis. And whenever there is a decrease in the platelet count, the condition is known as thrombocytopenia.